currently affiliated to School of Life Sciences, Jawaharlal Nehru University. He has a PhD in cancer biology from JNU. He has received a postdoctoral trainee award for prostate cancer research from the Department of Defense, USA. He has also received ICMR International Fellowship for Young Biomedical Scientists to visit University of California. He was assistant professor at the Department of Pharmaceutical Sciences, University of Colorado. He has received Indo-US Science Technology Forum 2019 award for establishing Center for Integrative Cancer Biology and Therapeutics. He has also worked as professor at the School of Life Sciences in the Central University of Gujarat. His areas of research interest are carcinogenesis, cancer chemo prevention and therapeutics, tumor angiogenesis, radiation therapy, anti-cancer drug development from natural agents, steroids and cancer stem cells. I welcome you, sir, to kindly deliver your talk.
Thank you. Thank you very much for the introduction on both the chairs, and also very much thankful to the organizer, especially Professor Guwahati, for giving me this opportunity to discuss our work. So what I am going to talk about is just a small work done by a PhD student in my lab, and she was working on radiation therapy, and also she wanted to know that if there is any compound, whether it is going to have any radiosensitizing effect or not, and if it is, then what would be the possible mechanism. So I will just focus on that aspect, and for that she took the example of prostate cancer. Since we work in several cancer models, for this prostate cancer was selected. You already know about the disease progression with respect to cancer. It starts from normal prostate epithelial cells, then thin adenocarcinoma. Usually when this prostate cancer is diagnosed, the therapeutic options what we have, you can see here androgen or androgen receptor targeting therapies. But what happens after a few years, the prostate cancer regrows, and at that time those cells, they become either androgen independent, it means they are not responding to the therapy, or they will have autocrine growth factor receptor signaling, and at the same time deregulated cell cycle progressions. And there are some other mechanisms that have been observed. But at this time, the intervention what clinicians are going to do for the patient is either radiation therapy or chemotherapy. So here, with respect to radiotherapy, there are some challenges. Development of resistance is one, and the toxicity is another one. These are two major challenges. They depend upon the sensitivity of the tumor tissue as well as the normal tissue. And therefore, the successful outcome depends what kind of cancer cells and what kind of dose is being used and how the patient is responding to the treatment. So for the study, we took one simple compound. This is silibinin, flavonide compound, on which we have worked a lot. This is just a structure showing hydrophilic and lipophilic portion of the molecule, suggesting that this molecule, if it enters into the cell, it can have a range of interaction, starting from extreme hydrophilic to lipophilic. So we did one experiment. This is a thymic nude mouse model. The advantage of this is that you can grow human tumors by using human cancer cell lines. So what you see here on the left hand side, we have controlled tumors. You can see they are bigger as compared to the other side, in which the silibinin was given in diet, and the treatment went on for 8 weeks. It was only 0.5% weight by weight. So So this experiment suggested that this compound may have anti-tumor effect against prostate cancer. And next, we wanted to know what could be the possible target of this compound. We did one simple experiment. This is clonogenic acid. You can see the doses 2, 5, 7.5 and 10 micromolar of silverine. This is 9L rat glioma cell line, which does not express epidermal growth factor receptor. And we expressed human EGFR in the cell line. If you look at all these wells, this drug is not showing any effect. The number of colonies, you can see, it is same. So once the EGFR was expressed here in this 9L rat glioma cell line, you can see the strong effect of this compound when the dose is increasing. This A431, this is skin squamous cell carcinoma cell line, which is known for high expression of EGF receptor. This is also showing almost similar trend. Then we thought that most likely this compound might be working through EGF receptor, and you might be also knowing that EGF receptor is kind of deregulated in almost 50% of cancer cells, cancer types as well. If you go for it's a signaling pattern, this is a classical activation of epidermal growth factor receptor through ligand mediated. You have EGF, TGF-alpha and pyridine. However, you might be also knowing about ligand independent activation of this receptor. This is stress-induced activation, where even ultraviolet radiation, ionizing radiation or some other stresses can also activate this receptor to transduce myogenic and survival signaling. We came across a paper in 2001 where it was first time reported that EGF receptor, this is a very big molecule, 170 kilodalton. So this molecule was found to be in the nucleus. When the scientists, they got surprised how a big molecule which is supposed to work on the cell membrane to transmit the signaling, why it is going into the nucleus. At that time, they visualized that if such a molecule is entering into the nucleus and going to work as a transcription factor, the whole concept of signaling is going to change. However, later on it was found out that EGF receptor is not having any DNA binding domain. So it was ruled out that this is going to work as a transcription factor. It means if it is going into the nucleus, then it has to have some functions. So that was the puzzle when people started working on. So on the left hand side, this is the classical signaling of EGF receptor, and it can influence almost all the processes of cancer development: tumorigenesis, proliferation, metastasis, chemoresistance, or radioresistance. On the right hand side, if you look at nuclear EGF receptor, we focus on this one. And this receptor is coming into the nucleus, and what could be the possible outcome? Then we focus on the last one, whether this is having any role in radioresistance or not. With this aim, we looked at the literature and found out that in clinical samples as well, if the patients are going under chemotherapy or radiotherapy, the presence of EGF receptor is there in the nucleus as well. However, there were no reports in prostate cancer. Then we did some simple experiment here. We did prostate cancer cell lines exposed to the ionizing radiation. And first we did clonogenic acid, G145 advanced prostate cancer cell line, BC3. You can see the dark line. This dark line is for only radiation treatment, and this less darker one is radiation plus silicon. In both the cases, this less dark line is below, indicating that colony formation efficacy is getting decreased in the presence of silicon. This is another cell line from lung cancer. Just we wanted to see whether it is there only for prostate cancer or also for other cancer. Similar result was there also for lung cancer cell line, A509. The more interesting observation was here, human embryonic kidney cell line, which is considered as non-neoplastic, non-cancerous. And here the result was exactly opposite. You can see that the dark line is below, and with the radiation plus drug combination, it is above, showing that in the normal cell line, this compound is having a protective effect. So here we got a clue that there is a selectivity. Nowadays, this is what everybody is looking for, an anti-cancer drug, how to make it selective. Cancer cell versus normal cell. That is what we observed here with this compound. Then we did some experiments related to cell cycle progression. In this you will find that in combination, that is black bar in all the cases. GTO phase arrest was there. Mid cell lines, some molecules which are associated with GTO phase arrest, like in B1, CDC2, CDC25C, all the molecules changed in that favor for arresting GTO phase. So that is the summary of this. How CDK cyclin gluon complex, if you inhibit, you get GTO phase arrest. And that was happening with this combination, ionizing radiation and siliconin. This is a very important point over here. If you block it here, cells are not going to enter into the mitosis or the cell division phase. Then for the selectivity, we just had to look at reactive oxygen formation. What is the status in this cell? And here again we got very interesting result. Just look at the last figure, figure D. First bar control, the next one is only siliconin, third one is only radiation, fourth one is the combination of radiation and the drug. You see, the reactive oxygen species in case of cancer cell line, that is G145, ROS level is increasing in combination. But when you go to the normal cell line, the result is very drastic. You don't see the increase in ROS, but ROS is being decreased in the combination. Another important observation here, you will find the third bar. If you look at the radiation treatment, the third bar is radiation treatment. In both the cases, the bar height is same, whether it is cancer cell or normal cell. So this tells that radiation does not discriminate between cancer cell or normal cell. ROS level produced is same in both the cases. However, the combination of drug is differentiating, and that could be a possible mechanism for protection of the normal cells.
Both these cell line expresses EGFR same level?
Same level I cannot say, but EGFR is there. Expression is there. So some more experiments were done related to apoptosis of cell death, and we find more cell death in the combination as well. This is related to checkpoint. So checkpoint inhibition was also observed. You see check1, check2 phosphorylation in the last treatment, that is very much reduced. I will come to the main point of the study where we are looking for EGF receptor. How the drug is modulating the translocation of EGF receptor. This is G145 cell, cancer cell line. The blue color is nucleus, Doppler staining. Red color is for EGFR, this is merged. If you look at only drug treatment, still the red color is outside of the nucleus. There is no overlapping of the red and blue color. But when the cells are exposed to radiation, you can see all the red color, it is coming into the nucleus. And also overlapping of blue and red. So this tells directly that EGF receptor is getting translocated into the nucleus once you expose the cells with the radiation. When we combine the radiation with the drug, then you see it's very clear nucleus. Again, the red color is outside of the nucleus. So here you can conclude that the drug is blocking the translocation of EGF receptor into the nucleus. So those are the conclusions. Just to support that, in large image with the radiation, EGFR in the nucleus, radiation plus siliconin, EGFR is outside of the nucleus. Again, you need to substantiate your results by doing Western, make fractions, cytosolic and nuclear, and show that where EGFR is located. So you can see here in the radiation 5-way, in the cytoplasmic fraction you don't have. Here in the nucleus you have, but when you combine, then you have positive result. Here in the nucleus, you don't have EGFR nucleation. Now next, since our hypothesis was related to DNA repair, we also had to look at DNA PK, DNA dependent protein kinase, which is very important for DNA repair. Here red color will represent the active form of DNA PK, that is serine 2056 phosphorylation. Green is again nucleus. Control and siliconin treatment alone, you will find similar observation. With the radiation, again you see red color is going into the nucleus. This tells that when you damage the DNA with the radiation, the DNA PK is entering into the nucleus to repair the DNA. And that is the mechanism how it usually happens in all the cells. But when we combine the drug with the radiation, again you can see the green colors, very green. There is no overlapping. So this tells that the drug is also blocking the nuclear translocation of DNA PK. In other words, if you combine both the regions, EGFR as well as DNA PK, the translocation was blocked by the drug which was being induced by the radiation. Now, this is again very important data related to the DNA damage in the form of STX foci you can measure. If you look at this bar diagram, the black one is 6 hours and next less dark one is 12 hours. So whenever DNA damage happens, immediately DNA repair machinery will start in the cell, they will start repairing the DNA. As a function of time, you can follow or see the kinetics of DNA repair, the treatment versus non-treatment, how it is happening. So if you look at this 6 hours versus 12 hours, in the 5 grade, there is decrease of DNA foci, means most of the DNA damage was repaired. In the combination, 5 grade plus 11, you can see this decrease is very less as compared to this one. So in 6 hours, the amount of DNA repaired in the drug treatment after UV exposure is very less. So in other words, in the cancer cells, when the drug is there, the DNA repair might be getting impaired, it is not efficient. And when the DNA damage is there, then likely what we expect, the cells are going for death or apoptosis. Next question you will ask, let me just summarize this, how it might be happening. So EGF receptor and DNA PK with only ionizing radiation, they are entering into the nucleus, and wherever DNA damage or stand breaks are there, they are going to repair. In case of TKVT, there is very well defined mechanism that when we combine radiation plus silicon, both the molecules, they are not coming into the nucleus, then there will be deficient repair, and corresponding we are expecting that there will be more and more cell death. The next question will come whether it is similar in the normal cells or it is also happening same mechanism what is there in the cancer cells. It means that this mechanism is different in cancer versus normal, that is same. So we took keratinocytes here as a normal cell. Did the same experiment. Here for EGFR you can see. The red color, again it is in the nucleus. In the normal cell, it tells that DNA repair mechanism is there and EGFR might be playing role. But when we combined with the radiation, there is still EGFR is in the nucleus. However, this result was very different in the cancer cell. So there is no blockage. So this tells that if DNA damage is there in the normal cell, this mechanism will operate for the DNA repair. That could be again a protective mechanism for the normal cell. So we just try to do one in vivo study to see whether similar things are happening or not in vivo condition. This is vaccinated doses as the patients are treated in the clinic. Total 15 gray dose we gave and the same cells were genografted. If you look at the kinetics of tumor growth here, tumor volume, blue line is for control, tumor is growing, and you just focus on this pink line, so radiation plus the drug. You can see this line is going down, indicating that tumor is regressing in the combination. So very strong effect. This is at the end tumor weight. It is again very significant effect. In terms of systemic toxicity, body weight, as you already know that radiation has very strong side effect. So this red line indicating the body weight loss after radiation treatment up to 15 gray. Pink line, which is combination, it's showing recovery in the body weight loss. And also diet consumption is getting recovered. Another important parameter is immunotoxicity, and radiation is given to the patients. Immune toxicity is a major concern. Again, if you look at the third bar in all the cases, ionizing radiation, total WBC count, lymphocyte count, platelet counts, they are getting decreased. But in the last bar, the combination is there, everything is coming back to the control. So immune protection is also there in the combination. We also did some immunohistochemistry just to look at the tumors that is happening. This is related to proliferation. This is related to H2X DNA damage apoptosis. All these results were in the favor of the combination. So now I'll just summarize whatever I have discussed. EGF receptor, which is on the cell membrane. This is its classical mechanism how it transmits signaling for growth and proliferation of cells. What we observed here, that EGF receptor is getting internalized into the nucleus, and in the nucleus it has role for enhanced DNA repair. On one hand, we are trying to damage the DNA in the cancer cell to kill them, but this is the resistance mechanism. EGF receptor is coming and trying to repair the DNA, and the cell will survive. It was again supported by the translocation of DNA decay, that yes, both are coming to the nucleus, and DNA repair will happen in the cancer cells. They are not going to die. This is the resistance mechanism. And what this drug is doing, once we put this cell along with radiation, block the nuclear translocation of both these receptors as well as DNA decay, and then these cells become radiosensitized. That is how you got more and more cell death in the cancer cells. So this is my group in the laboratory. There are students who are working on different projects. However, for this project, as I said in the beginning, this was a PhD work of one of my students, Hane Namya. And for this work, she also got Fulbright fellowship. She went to University of Colorado, did some work over there. She was also selected for this Nobel meeting in Germany, and then went for postdoc. Currently, she is working as instructor in the Radiation Oncology Department in Stanford. Funding was given by DST and LC. So this, thank you all for listening. And if there is any question.
Have you used radio-resistant cell line to see the effect of that?
Sir, we have 3 cell lines, LNCAP, PC3 and D145. So first we did the experiment to find out which one is more radioresistant. So D145 is the strongest in this term. It's a highly radioresistant cell line. And that is how it was used.
Dr. it's a good piece of work. A couple of questions. I think first concept that nuclear translocation of EGFR by I think means that Chinese life from India published much of paper including cancer cell receptor started around 2000. But my question to you, using your cerebellum and response to that radio resistance, most important point, when there is a sapling of EGFR, what is being a RTK, receptor tyrosine kinase, what is the status of tyrosine phosphorylation of EGFR when it is translocating into the nucleus?
So usually, sir, there are a lot of studies on that, and we are also